have actually been published, particularly an explosion over the last decade where people are really interested in being able to understand how tumor cells and myeloid cells interact to promote angiogenesis in tumor microenvironments, and how people might actually try to inhibit this kind of response to try to inhibit the growth of the tumor cells. Much less actually has been learned about how the endothelium regenerates itself or repairs itself within the vasculature. And about a decade ago, we became very interested in some very rare cells that circulate in the blood that are endothelial cells. And we learned that those cells had the capacity in the circulation to bind to things that were in the circulation, such as artificial materials when they're implanted, or when patients get hooked up to cardiovascular devices, that you can actually generate from what's called fallout endothelialization, an endothelial coating on those devices. So we became interested in these circulating endothelial cells and wanted to learn more about their properties, their ability to be expanded, and think about what we might do with them therapeutically. So we took human adult blood, and we took also umbilical cord blood, plated the cells, took the cells that adhered, and over a period of time, one would see these kinds of colonies emerge on the well. One of the things we noticed right away is that some of the colonies were very small and some of the colonies were much larger, suggesting to us that there was heterogeneity in the proliferative potential of the cells that were forming these colonies. And we thought probably the only way we were going to learn about those differences that were intrinsic were to actually replate these colonies at a clonal level. So we developed a clonal culture system where we could actually take a colony. When we first started out, we actually used to label them with retroviruses encoding GFP so we could more easily sort them. We would plate then a single cell per well under defined conditions, and then we would score the growth of the colonies over a 14-day period. And what we would see is that many of the wells contained the single cell that we loaded. Some would contain small colonies that we actually called clusters. There were only 2 to 50 cells. And then there were larger colonies that appeared, and then there were the very large colonies. And the one thing we learned about this hierarchy is that these colonies, the largest colonies, contained within them cells that upon replating would give rise to colonies of the same size or greater. However, if we replated any of these colonies, they could never actually give rise to a colony of the same size or bigger. So these colonies that we called high proliferative potential actually displayed features of self-renewal potential, suggesting that there was unique populations that were actually replicating themselves going forward. The other property that we were very excited about was to take these cells, to implant them in this case, collagen and in some cases fibronectin containing gels, and then implant them subcutaneously into immunodeficient animals, and then allow the implants to go over a period of time from 2 to 4 weeks, all the way out. The longest we've ever looked at is 9 months. And then to image those vascularized implants. And what you see is that the human cells in the collagen form vessels. This is a antibody against human CD31, and you can show a nice capillary-like structure. And inside this are mouse red blood cells. If you inject lectin intravenously, you can see that the human cells have actually inosculated with the murine circulation, and now The intravenously injected lectin is actually labeling the intraluminal side of the vasculature. So we were very excited. These cells not only displayed clonal proliferative potential, but they also intrinsically had in vivo vascularization potential. The difficulties with working with a human cord blood, though long term, is of course it's a one and done sort of phenomenon. You isolate it and then you got to move on to the next patient, the next patient, the next patient. So we wondered, are there protocols that we could differentiate human iPSCs into endothelial cells that display the same kind of cord blood ECFC-like properties? So people typically, to look for endothelium that's differentiated from IPS, have a list of things they look at. They look at cell surface antigens, immunofluorescence, PCR. They try to test their ability to form tubes in matrigel, in vitro and in vivo, and a few other assays. But what we were looking for were cells that would have not only the phenotypic characterization of an endothelial cell, but it would have the same proliferative potential as cord blood, and it would form vessels in vivo when we implanted them. So I'm going to just cut to the chase, and we published a paper a few years ago where we actually derived a protocol that would allow us to find these cells. We start out with human ES or IPS cells, then MTZR1 media. We then actually take that media off, give them a pulse of activin A for one 24-hour period in the presence of FGF2, BMP4, and VEGF165. All of this, these three growth factors continue for the next 10 days, and at day 12, you can see the appearance of a very important subpopulation that is neuropilin-1 positive and CD31 positive. These cells, in addition to expressing neuropilin-1, also express CD144 and CD34. But it's key that you pick out this particular subset if you want to find the ECFCs downstream. We then sort this population, and over the next 7 days expand it up, and we switch them from a stem line 2 serum-free media system to an EGM2 based system. And as I said, they express 144, 31, they also express 34, they express 146, and a variety of other molecules similar to the cord blood cells. They almost all express neuropilin-1, and they're all KDR positive. When you look at the cells in monolayer culture, they form a nice cobblestone layer that's exemplified here. This is a neuropilin-1 and CD31 stain, and the CD31 is very nicely interendothelial in its nature. If you put them on top of a 2D matrigel culture system, they'll actually form luminized structures. But most importantly, if you implant them in vivo, they'll make human blood vessels in these collagen implants in the mice. And importantly, when you plate them at the single cell level with cord blood cells in pink, human ES H9 cells in green, and human IPS in blue, you'll see that they have a very similar distribution of proliferative potentials to the human cord blood cells. So we were very excited that this particular protocol done in this way gives you this population of cells. We're also very excited to see that the cells do inosculate in vivo. So if we have this image of an implant, in blue is the mouse circulation with a 31 specific antibody against mouse CD31. And in red were the human IPS TD tomato expressing cells. And you can see how the cells actually inosculate. They actually wrap around the mouse vessels and then penetrate the mouse vessel to actually abscond with the blood from the mouse vessel into the human circulation. And they do this very quickly. So it takes them about 5 days in vivo, and they will connect with the circulation. And over the period of the next 3 to 4 days, they solidify the blood flow distribution into the vasculature. If you look at gene expression, we just have here just a quick summary of some of the genes. Obviously, they're pluripotent genes that actually make a network that you can see that are in the uncultured or the undifferentiated iPS cells. But the important thing is if you look here at the endothelial genes that pop up, you see there's a very close correlation between the human iPS, the human ES, and the cord blood cells in terms of gene expression parameters. The important thing is, can you make sufficient numbers of these cells to actually think about using them as a therapeutic? So we did one long-term study where we took 10,000 pluripotent stem cells on day 1. And by day 12, when we isolated the neuropilin-1 CD31 population, that particular population had expanded to 30,000 cells. And then as we passaged the cells over the next few weeks, this number expanded such that by day 61, we went from 30,000 cells to 1.1 trillion cells. So these cells possess an amazing proliferative potential in vitro in this expansion protocol. They're not immortalized. They do eventually senesce. After about 15 to 18 passages, they'll upregulate typical senescence genes and they'll stop proliferating. So they're real cells, non-transformed, but cells that are rather unique to the endothelium in possessing this kind of proliferative potential. So if you look at the kind of expansion you can get with most endothelial differentiation protocols, you can maybe get 1 to 11 endothelial cells per input IPS. But here we got basically 1 to 10 to the 8th of these cells from a single starting pluripotent stem cell. So clearly you can get enough cells in this growth phase that you can consider patient treatment. So just to summarize this part, the endothelial lineage differentiation of human iPS in our ECFC protocol results in generation of a small subset that exhibit umbilical cord blood-like ECFC phenotype and function. Now the question is, what might they do if you implant them in a hypoxic ischemic environment? So I'm actually a neonatologist and I work in the newborn intensive care unit. So I've worked all my career with these prematurely born babies. We're doing really much better in their overall survival, but one problem that remains is these babies get a retinopathy of prematurity, which is a very bad disease and still can cause the loss of sight in some infants. And it's caused by the fact that the retina is just undergoing very nice vascularization when many of these babies are inappropriately, abruptly born. And with the loss of the placenta and the loss of the maternal nourishment, several key factors have been identified that actually plummet at the time of birth of these babies. And in addition to that, you're taking them from an environment in utero where the oxygen tension is relatively low, and you're bringing them out into an environment where oxygen is high. And this actually causes further pruning of the vessels because they don't need to be present because there's a high oxygen tension. Unfortunately, what happens then with the loss of all of this in the context of a high oxygen is once the baby starts to grow, then unfortunately this tissue becomes hypoxic and ischemic, and you get a massive neovascularization that occurs that's very inappropriate. It grows actually away from the retina into the vitreous and can cause bleeding and detachment of the retina. So to try to avoid this, we try to do intravenous nutrition in these babies. We try to control those oxygen tensions so that we don't have them any higher than they possibly have to be. And then when you get to this phase, the only real good rescue therapies are either laser ablation or the use of anti-VEGF intravenous injected drugs. Now the mouse born at day 1 has almost no retinal vascularization, but over the first week of life completely vascularizes this structure. So it's a wonderful model to actually study retinal development postnatally in the animal. And in fact, if you expose them to high oxygen concentrations for a brief pulse after the first week of life, they'll actually go through the same stages of retinopathy that you see in human infants on an abbreviated scale. So we actually used this model that was started by Lois Smith, and we've actually examined the infusion of 100,000 of these iPS-derived ECFC or vehicle control, where the cells are actually administered right into the jelly-like vitreal substance. And what we saw in the vehicle-controlled animals, as expected, there was massive neovascular tufting. So instead of forming a nice capillary bed along the base of the retina, these proliferative masses of endothelium grow out toward the vitreous. But in the animals that had received the ECFC injections, you had a much more well-developed retinal vasculature with a single neovascular tuft seen here. Also importantly, in the vehicle-controlled animals, there were regions that remained avascular. And in the ECFC-injected animals, there was actually a 36% reduction in this avascular area. So clearly in this retinopathy model system, the injection of these cells actually prevents retinopathy. So it's a little bit of a surprise. By giving cells that form vessels, you can prevent overvascularization. And the key is, we've learned over time, is that the human cells that we inject purposefully migrate into the hypoxic ischemic area. And they make their own vasculature, which is shown here. So here is a composite image. Here's all the nuclei. Here in the yellow color is the green ECFCs. The red is a lectin injection. And you can see the human cells have actually integrated into the retinal vasculature. And in so doing, they relieve the hypoxia and the ischemia and prevent the emergence of the neovascularization. So this is an exciting finding. In a second model I'll quickly tell you about, which is the hind limb ischemia model, you simply remove the femoral artery in immunodeficient mice, and then you look at the ability of your test substance to rescue blood flow to the limb and help the limb repair itself. And in this model system, if you use a vehicle and you inject it intramuscularly, you really can't salvage any of the limbs. And 1 out of 10 have foot necrosis. 9 out of 10 actually lose the limb in this model. However, if we gave a single injection of a million cells intramuscularly, these human ECFCs, in this case, 6 out of 10 were rescued. 1 had a foot necrosis, and 3 out of 10 still had limb loss. But this was with 1 single injection. And it was exactly the same kind of result that we obtained with the cord blood cells. So this just depicts this same data on another scale, and you can see there's a significant enhancement of recovery. And this shows the limb is completely restored with its blood flow as compared to the control animals, which lose their limb. So these differentiated human iPS-derived ECFCs significantly rescue blood flow to ischemic limbs. So the exciting thing is with this protocol, we can expand up this population of cells, and in 2 preclinical animal models of human disease, we can show there's significant enhancement of the vascular repair. So how can we potentially use these cells now? Well, in addition to ongoing preclinical development of these cells, Axol is actually selling these cells. You can find their information there. And there's a whole host of uses that you can anticipate. You can screen for angiogenic or anti-angiogenic molecules. You can actually take the ECFCs, put them into a 3D collagen matrix, and actually allow them to make the intravascular luminized structures, and then add drugs to see what you can do to disrupt the luminized vessels that have developed. You can also do 3D screens to look at ECFC stromal cell interactions, ECFC tumor cell interactions. There are a lot of folks who are pre-vascularizing tumor constructs using these cells before they're implanted and looking at how that modulates the tumor growth. They're, because they have such high proliferative and expansive potential, you can actually do a gene regulatory network analysis, so you can knock things out, put things in, and look at downstream how that influences the 3D vascularization, which we really like as a sort of a preliminary way to find out which are the most active compounds. Then you can actually go to the in vivo studies in the animal, and we find that the 3D luminized vessel formation in vitro is a very good predictor of whether or not you're going to get vessels in vivo. And you can also think about delivering particular molecules that you have of interest, where you want to know whether your protein or your peptide or whatever you want to make is actually working in the mouse system. You can actually express it in these cells. And since these cells automatically make a vasculature and they hook up to it, they'll pump out your product in a continuous way, and you can actually test what your product is doing in the animal cells in vitro. So that's what I was going to say, and I'll just leave this up, but I'll take any questions if there are questions.
So in your assays where you have the connection of the vasculature, how do these ones compare to mature endothelial cells?
So if you take the earliest passage HUVECs that are the most proliferative, they'll have similar activity to these cells. But the problem is getting those early P2 HUVECs. You got to keep doing it and doing it and doing it. So they're very similar. And I don't know if you've ever isolated HUVECs. One of the things you notice is when you take the HUVECs and you plate them, 2 hours later, most all of the cells actually will go down and adhere to the bottom of the plate, and it looks like you've got a wonderful transfer from the vessel onto the plate. The disappointing thing is when you come back the next morning, the vast majority of the cells actually release from the plate and have died. And all that remains behind is a small number of cells that are attached to the bottom of the plate. But those cells actually are the cells that give rise then to the rest of the HUVECs that emerge. So in fact, we're trying to prove this, but we believe, in fact, that the cells that we are showing here that we have identified from the umbilical cord blood, those cells are actually coming from resident endothelial colony forming cells that live in vessels. So in fact, those cells would be equal. So we're still trying to give evidence for that. Yes?
So, Ray Valdez is one
So in the hind limb ischemia, we've never gone out longer than about 6 weeks, but they do contribute to arterial vessels, to microvessels and veins. In the implant model, we've gone out to 9 months, and there's no teratomas. We've done, I don't really remember the number, it's more than 40 animals, and we've gone out 9 months and not had any teratoma formation, and they remained a part of the vasculature of the animal. The interesting thing to remember about, you know this, the vascular endothelium never is a very efficient system. So if you need to prune the system, you'll prune it. So when we put these subcutaneous implants into an animal, we're putting them in a place that doesn't really need vasculature. But it forms anyways, and so we can find a vascular that persists long term, but it generally wanes with time. If you put it into a physiologic space that actually needs vasculature, like the retina, so in those retinal studies, we actually could see the integrated green human cells in the retina just by looking with a scope into the eye of the animal. You can actually see the green vessels that have formed in the retina. And those persisted for 5 months. So we suspect in the femoral model they would persist longer. We just haven't done the long-term study. Yes?
My impression is that Cuba cultures, while the idea that they can contain progenitors has been around for a long time, but those cultures can't or aren't usually maintained for a very long time.
That's correct.
Do you think that's a technical issue, that people just haven't used the medium, the approaches, and the splitting protocols that you would use for that? Or is that an argument that these cells really are different in some way?
Yeah, we're working very hard on that. We've been trying for years to find a unique marker of these cells that would allow us to actually go back and see them in a vessel if they are in a vessel. And while we are not quite there yet in the human system, we have been able to do this in the mouse. And we have a paper submitted on this where we actually can identify the colony-forming cell with specific cell surface antigens in the vessels of the mouse, in the arteries, the microvasculature, and the veins. And we can do with a fate mapping study that if you transplant those cells, they actually give rise to cells that are able to regenerate the vasculature in an injury model. So using these kinds of tools, we hope to be able to learn this intimate relationship between cells that are in the endothelium of a vessel that serve as sort of the stem and progenitor cells of the endothelium, and how they make the circulating population.
So those earliest, or those cells, I take it, there is a marker AC-133.
Yeah.
Is that human only, or?
That actually isn't on these cells, and it's not on the mouse cells.
So what about the idea that the cells that you are talking about, do they have hemogenic?
They're not hemogenic. So these are actually stem and progenitors only for the endothelial lineage. These are not hemogenic.
So do those cells differentiate to other tissue-specific endothelial cells, such as liver endothelium?
So actually, we've identified this population in brain, liver, lung, heart, and bone marrow to start with. And the frequency is different in all of those tissues. The localization of where those stem cells reside changes as the animals age differently in the different organs. And so we're now actually in a big project to identify the gene expression patterns of those different populations over time. We anticipate that the vasculature basically sets out as a basic fundamental path that then gets modulated by its microenvironment during organogenesis. and takes on tissue-specific properties. But the fundamental property of making arteries, veins, and capillaries actually is the same in all the tissues. It just gets fine-tuned as those tissues are changed.
Can I ask your ECFC mimic the cord blood?
Mm-hmm.
Do you think it's the same as adult circulation?
Ah, good question. So I think their ECFCs in adult peripheral blood, they're just far fewer, and they seem to have lower proliferative potential. They don't expand as much.
So thank you very much. I think we have to entertain her. Sorry, Professor Yogi is around for a few more questions, so a little bit longer. So if we could thank him once again. Thank you all for coming this morning. Thank you.